Hi guys, this is the female reproductive system of chapter 22. So we're going to get started with looking at the female reproductive organs. So this is the overview. We have the vagina, which is a fibromuscular tube that is going to lead to the uterus. The most inferior portion of the uterus is the cervix. The cervix contains an opening, or it has an opening. That opening was called the os. And then we have the organ itself. The myometrium is the smooth muscle of the uterus. The endometrium is the interior lining. And then on top it's the perimetrium, but we won't worry about that. We can see that our gonads, which are the ovaries, are going to create the ovum or the gametes. When the mature oocyte is produced and ovulated, it's going to be transported into the infundibulum of the uterine tube. The infundibulum has these fimbria. They are finger-like projections on the ends of it, and they are to help coax the egg into the infundibulum portion of the uterine tube. And then there the egg is going to travel, I should say ovum, and possibly implant into the wall of the uterus if she's pregnant. So we're going to spend a lot of time looking at the ovary, and the book immediately goes into granulosa cells. So looking at your outline, inside the ovary there are special cells called granulosa cells, and these are the cells that are really going to take care of and mature and have a large role in maturing and producing the oocyte. So just a quick comparison between the Sertoli cells of the male and granulosa cells of the female, because they are pretty much homologous cells. So they're both going to surround the developing gametes. Remember that the Sertoli cells are secreting fluid into the lumen of the seminiferous tubules to support and also to transport the sperm. And the granulosa cells are going to secrete antral fluid, so the fluid that's going to eventually create the antrum of the follicle. The Sertoli cells provided nutrients for the developing sperm, and so do the granulosa cells. They are going to do so by actually creating gap junctions that lead from the blood supply into the oocyte. We'll talk about that in a bit. They're going to secrete molecules that are going to stimulate spermatogenesis, and granulosa cells also secrete molecules that support the follicle development. They both are going to secrete inhibin that inhibits the follicle stimulating hormone. This is from embryonic fetal life, right? So they're going to secrete this Mullerian inhibiting substance. These guys are going to secrete estrogens. The Sertoli cells, remember, form that blood-testis barrier, and the granulosa cells also form a barrier around the oocyte, and that's called the zona pellucida. All right, and then lastly, just they're secreting some antigen-binding protein, which the granulosa cells do not, but they do secrete estrogen. All right, so moving on. We'll talk about the progression of the granulosa cells more in detail as we look at the ovarian cycle. This is just a quick review again of the female reproductive tract. So the vulva involve the labia majus or labia majora. The labia minus are the smaller folds of skin for forming the external genitalia. Then we have the vagina here, right? So it's a fibromuscular tube. The cervix is the inferior portion of the uterus. Here we have the uterus, which of course receives the fertilized egg and houses the growing embryo and fetus. Our ovaries are the gonads. The uterine tubes are going to receive and propel the oocyte and push it towards the uterus. And then that's about it. So we have some erectile tissue here, the clitoris, which is homologous to the erectile tissue of the penis, and then some glands are not shown here. So just a quick review of the female reproductive system. This is the vulva, right, the external genitalia. So here's the clitoris, again, homologous to the penis, the labia majus, labia minus. The hymen, which is not shown here, is going to be a membrane that covers up the opening to the vagina. So that membrane is variable in women. Some women have a very tough, thick hymen that is, it bleeds when it's broken. And then some women have a barely there, very thin hymen that's barely noticeable if it's ruptured or broken. Opening to the vagina, opening to the urethra, and the mons pubis is a pad of fat that's found over the pubic symphysis. All right, so let's look at the ova and their development. So the development of ova is called oogenesis, and it's very similar to spermatogenesis. But it's also different. So we're going to have to look at the differences. In the fetus, so in utero, we have meiosis occurring. So we have oogenesis occurring, which is different than the male, because male, the spermatogenesis does not start until puberty. So we have oogonia, which is our stem cells, which are 2N, and they're going to start to undergo differentiation and mitosis, and they're going to differentiate into primary oocytes. So this is really similar to the male, right? We had a spermatogonium and primary spermatocytes. So what's a little bit different is that the primary oocytes are going to be arrested in right as they enter meiosis 1, they're going to get arrested, and this is called a meiotic arrest. So the stem cells, these are actually still 2N, these primary oocytes, because they haven't completed meiosis 1, which would actually create a haploid cell. So they're still 2N, and they're arrested. And so oogonia, there's a lot of these guys. There are 2 to 4 million clones in the ovary, so 2 to 4 million of these guys. Okay. And then they're going to all sort of differentiate into primary oocytes, but they're not all going to arrest in meiosis. So we're going to lose a lot of these oocytes because they're not going to be arrested and they're going to degenerate. So the primary oocytes that don't undergo meiotic arrest will undergo apoptosis. And if apoptosis is a new word for you, it's programmed cell death. So apoptosis is programmed cell death. So it's a way to die in a neat manner, and so it's not to create sort of inflammation or alert the immune system with lysis. Okay, so then when the baby girl's born, there's about 1 to 2 million primary oocytes. So 1 to 2 million of these primary oocytes. So a baby girl is born already primed with all the primary oocytes that she'll ever need in her lifetime. Out of these 1 to 2 million, only about I think the number is about 400 to 500 ever mature and go through, ovulation. Okay, so when the menstrual cycle starts, okay, we're going to have secondary oocytes form. So these arrested primary oocytes are in her ovary all through adolescence, and then through the hormonal changes of puberty, then we start to have a maturation of these primary oocytes. They're going to go on to form secondary oocytes. And secondary oocytes are going to be basically the finished egg, and that's going to be ovulated. And then so the secondary oocyte actually never completes meiosis 2. So meiosis 2, the secondary oocyte's actually arrested in meiosis 2. And then only after fertilization does the secondary oocyte complete meiosis 2. So we'll look at that a little bit closer later, but just one thing to keep in mind, that in this flow of meiosis, there is not 4 gametes being made. So if you remember for spermatogenesis, we had 1 stem cell, which would divide into 2 cells, and then those 2 cells would undergo meiosis 2 and create 4 spermatids. Okay. In oogenesis, every time there's a meiotic split, we do not have an even division of cytoplasm, and only 1 cell gets the lion's share of the cytoplasm and goes on to complete the next stage of meiosis. The other cell is called a polar body and degenerates. So in this way, every time we have a meiotic split, we have 1 cell that's going to be functional and 1 cell that's not functional in the polar body, and so we only have 1 ovum. So at the end of oogenesis, we only have 1 mature ovum, whereas the end of spermatogenesis, we have 4 mature sperm. All right. So in terms of the next topic is a sexual response in females. So the vascular congestion of the clitoris. This again, the clitoris is erectile tissue. It actually contains the corpora cavernosa, which is the same tissue that you find in the penis. So it's a sexually sensitive and enlarges also with excitation. We also have vaginal secretions, nipple erection, and orgasm, which includes contractions of the vaginal wall, contractions of the uterus. So these are both smooth muscle contractions as well as skeletal muscle contractions and an increase in blood pressure and heart rate. This is the homology between the glans tissue of male and female erectile tissue. So you can see that the male in letter B is obviously larger and it's mostly external, whereas the clitoris is the same kind of erectile tissue, but it is mostly internal. So we have the glans, which is the end, the glans penis, and the glans clitoris. The glans is what is on the external surface of the female, but everything else is interior. All right, so let's get started with the menstrual cycle. So this is going to be a rather complicated cycle. It includes an overlap of three parts of a woman's body. We have the ovarian cycle, which we'll look at first. Then we have the ovarian cycle has two subphases, the follicular phase and the luteal phase. Then we have what's going on in the uterus, and there's three different phases there, menstrual, proliferative, and secretory. And then we have the fluctuations of hormones within this approximate 28-day menstrual cycle. And so we'll look at that overlap. So the menstrual cycle is about 28 days, but this varies between women. And of course, this is if you are a female, and this may look very, very familiar to you of what's going on in the menstrual island. All right, so let's take a look at the ovarian cycle. So what's going on in the ovary is going to be cut into half. Half of the woman's menstrual cycle will be involved with maturation of the follicle. So if I just drew a line sort of like through the ovary like that, this half is going to be called the follicular phase. Because what's happening here is that we're maturing the follicle. The follicles are starting off very small in a primordial follicle, then they're growing to a primary follicle, secondary follicle. What ends the follicular phase is ovulation. So when there is ovulation, we are shifting over to what we call the luteal phase. And the luteal phase is called that because of a structure called the corpus luteum, which is formed right after ovulation. So this is just a quick overview of the two different phases of the ovarian cycle. So this is another way of looking at it. So the follicular phase, again, is with maturing the follicle. The luteal phase is where we have the corpus luteum, right? So this is the corpus luteum. And then what splits the two phases apart, right? The stimulus that shifts the phases is ovulation. And ovulation is the ovum leaving the follicle. All right, so let's take a look at this. We'll come back to this picture. And actually, I want to take a look at this image with you guys. So what's going on in the ovarian phase is that in the follicular phase, from menstruation to ovulation, we have follicle stimulating hormone and estrogens, which stimulate the follicle growth and development. So you want to be able to remember and recognize the differences between the primary follicle, secondary follicle, and tertiary follicle. So the primordial follicles are very, very small. They are created from simple squamous cells here, and they are housing that oocyte. So the primordial follicles are going to then start to mature. So there's going to be some that are recruited to mature, and they're going to form the primary follicle. The primary follicles, the cells become cuboidal, and they become there's mitosis coming, producing more and more cells. Those cells are going to now differentiate into granulosa cells. Those granulosa cells we talked about early in the talk. So we're going to create a stratified looking primary follicle. And then the granulosa cells are going to start to secrete the zona pellucida. So the zona pellucida is going to be a thick membrane that covers the oocyte, and the zona is not cellular, it's just carbohydrates and proteins. Let me actually show you the zona, right, in this picture here. So we have the oocyte in the center. The zona is that band of white around the oocyte. So it looks like it's empty, but it's not. It's just carbohydrates and proteins. It's a thick layer that covers the oocyte. So because the zona is this barrier between the oocyte itself and then other cells and the bloodstream, the granulosa cells actually all communicate with the oocyte by creating gap junctions, and they extend their cells, and they basically deliver nutrients to the oocyte with these extensions provided by these gap junctions. So it's pretty amazing. The secondary follicle, so let's just take a look at perhaps this picture, right? So this is our primordial follicle. This is our primary follicle. This is going to be our secondary follicle. So in a secondary follicle, what's happening is the granulosa cells on the outside, they're going to start to develop into thecal cells. And the thecal cells are going to start to secrete androgen, actually. They're going to secrete testosterone. And the testosterone is going to diffuse into the granulosa cells, where they're going to change the testosterone to estrogen. So the granulosa cells secrete estrogen, but it's the thecal cells that respond to the luteinizing hormone. The thecal cells are going to produce androgen, and then the androgen gets converted to estrogen in the granulosa cells. Okay, so we have our secondary follicle, and in this stage, there's going to be a winner of this race. So in the secondary follicle, they're not all going to mature to a tertiary follicle. In fact, only one is going to mature into a tertiary follicle. And the difference is, when we have a lot of these secondary follicles, there is a race to respond to our LH and FSH. So we're all getting this luteinizing hormone and follicle stimulating hormone. And what they're doing is they're going to start to build more receptors. So these hormones will prompt the cells to grow bigger and also to put up receptors on the surface for LH and FSH. So basically, the cell that grows the fastest and puts up the most receptors to LH and FSH will win the race. Because what's happening is, as these secondary oocytes grow, they are going to secrete estrogen. The estrogen levels are going to negatively regulate FSH and LH. Now, if you have more receptors on your surface to LH and FSH, then even if those levels go down, you're still going to be able to respond to LH and FSH just because you have more receptors and you're more sensitive to LH and FSH. A cell that does not have a lot of LH or FSH receptors, if those levels decrease, then you're going to be at a loss because you're not able to detect that decrease in concentration because you just don't have that many receptors for them. You're less sensitive to the amount of LH and FSH. So the ones with the fewer receptors will actually start to die because they're not going to be as responsive to LH and FSH. The secondary oocyte that has the most receptors to LH and FSH wins because as those LH and FSH concentration goes down, this secondary oocyte is still very sensitive to those levels, even at lower concentrations. So we sort of have this cell that won the race, and it's the only one that's going to go on and mature into a tertiary follicle. So once we have a tertiary follicle, also called a Graafian follicle, it's going to look like our number 4. So this is our Graafian follicle. So we have the difference between secondary and tertiary is that we have a fully formed antrum. So the antrum is this space within the tertiary follicle that's filled with follicular fluid, and it's really, really large, and it's basically going to enlarge, 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 and then it's going to ovulate. So what's also happening in the tertiary follicle is that we have a corona radiata developing. So the corona radiata is a band. So in this picture, it's orange. It's a band of the granulosa cells that are going to surround the secondary oocyte and ovulate, also be carried with it with ovulation. So when a secondary oocyte is ovulated, it's going to contain around it. So if I just blow this up, right, this is our secondary oocyte. Then we have a band of clear zona first around the oocyte, and then we're going to have all of our granulosa cells that are formed in our tertiary follicle, right? So this is called the corona radiata. And both the corona and zona are going to follow with the oocyte as it's ovulated. Okay, so what triggers ovulation? So let's go back to our picture here. All right, so what's happening here is that we just talked about what's happening in the ovary in the follicle development. So we have our primordial follicle, our first one, our primary follicle, a secondary follicle, and a tertiary follicle. What you see in terms of the hormone levels released from the anterior pituitary are gonadotropic hormone levels, LH and FSH. We see a gigantic surge in luteinizing hormone. Okay, so it's called a surge, this LH surge, and that's going to trigger ovulation. So the antral fluid that you see here with my X is going to allow the oocyte to detach from the wall and to float around in it. And then the Graafian follicle will move to the surface of the ovary and then push against the ovary. And then these enzymes will actually start to degrade the wall of the ovary and then release the secondary oocyte into basically where the fimbria are, and the fimbria will move to sort of sweep the egg into the fallopian tube. But it is this surge in luteinizing hormone that triggers ovulation. All right, so what happens? So in most cases, you have one ovulated egg every cycle. What happens if you have two? Then that is our fraternal twins. So if two ova are released with ovulation and they're both fertilized, then you get something called fraternal twins. So they are genetically different from each other because what's going on is we have two different eggs, right, with two different genetic profiles. We have two different sperm, again with different genetic profiles. And so we're creating basically siblings. That's how fraternal twins can be a boy and a girl, or two girls or two boys, but you're basically having a brother or sister at the same time. You're genetically different from each other, right? So this is a really interesting picture of basically you can see it's a white father and a half Jamaican mother. And then these are the fraternal twins they have. So they're very, very different. A lot of people mistaken them for being, of course, friends and not sisters because they look so different, but they're in fact fraternal twins. Identical twins, by the way, is different. Identical twins is where you have one ovum, one sperm, so you have one zygote being produced, and then in the process of developing or like dividing through mitotic divisions, there is kind of a split of these cells. And so when the one zygote, this ball of cells, splits, they become identical twins. Identical twins have the same genetic profile. Okay, it doesn't mean that they act the same or behave the same or think the same, but genetically, on the DNA level, they are exactly the same. So identical twins. So the word monozygotic and dizygotic are given to this. So with fraternal twins, because you have two zygotes being made, this is called a dizygotic twin. And with identical twins, because you only had one zygote being made, this is a monozygotic twin. All right, so those are our twins. So let's go back to this picture here. So we have our ovulation that's triggered by this drastic luteinizing hormone surge. Now that we are moving on to the luteal phase. So the luteal phase, again, it's named after the corpus luteum that forms first. So the granulosa cells and the thecal cells that are still sort of forming that follicle will then differentiate into the corpus luteum or luteal cells. So if you look at your notes under luteal phase, we have a ruptured follicle. It becomes a gland called the corpus luteum. The corpus luteum is going to secrete estrogens and progesterone. So progesterone is really, really important for keeping that functional endometrium lining thick. So we'll turn our attention here very quickly, but progesterone has a really good name because pro is for, gestation is pregnancy. So in order to have a successful pregnancy, the lining, the endometrium of the uterus has to be very thick, very nutritive. So progesterone is going to prompt that uterus, the lining, to be very, very thick. The corpus luteum is going to reach maximum activity about 10 to 14 days after ovulation, and then it starts to degenerate. So that degeneration is called luteolysis. When luteolysis occurs, you're going to have a drastic drop in estrogen and progesterone, of course, because the corpus luteum is what secretes estrogen and progesterone. So if you look at the corresponding levels of estrogen and progesterone, we can really see that the progesterone levels shoot up. Estrogen levels are still relatively high, but they do start to dip because what happens is there's a shift where the estrogen levels go down and progesterone levels rise. However, when the corpus luteum completely degenerates into a corpus albicans, which is basically the scar tissue of the corpus luteum, we see a drastic drop in both our progesterone and estrogen. And that drastic drop is going to trigger menstruation. Okay, so if the oocyte is not fertilized and there's no pregnancy, we get this degeneration of the corpus luteum after 10 days, and it becomes a corpus albicans. What that does is it drops the estrogen and progesterone, and that drop triggers menstruation. It triggers the menses, or the loss of the functional layer of the endometrium. However, if a woman does become pregnant, what happens is there's implantation that takes place, and then the developing placenta, so the placenta that is being formed will actually secrete a hormone called human chorionic gonadotropin, and that is going to act on the corpus luteum to stay intact. So there's no degeneration, so the corpus luteum can keep making estrogen and progesterone to keep the lining thick, to keep pregnancy, to keep the pregnancy. So it's the HCG that maintains the corpus luteum, and it maintains the estrogen and progesterone levels. So this actually comes into place, at that 10-day mark, or before the 10-day mark, so you can actually keep the corpus luteum intact. All right, so let's take a look at what's going on in the uterus. So we have 3 phases of the uterus. We have the menstrual phase, you can see here, where we actually are shedding the lining. And this marks day 1 of the menstrual cycle because it's the most obvious start point, right? Everything else is kind of invisible to the naked eye, but we know when a woman starts to bleed, right? So that's day 1. So it's triggered by a very drastic decrease in estrogen and progesterone if the woman is not pregnant, because the corpus luteum has degenerated. So that's your source of estrogen and progesterone. When that goes away, the tissue decreases, it dies, it sloughs off, and it's lost through the vagina, right? And that's called the menstrual flow. After that, we have the phase called the proliferative phase, because we need to rebuild and regrow the lining of the uterus for the next cycle. So what you see here is estrogen levels are going up. So what's really prompting this proliferative phase is estrogen. Okay, so estrogen is really, you can think of estrogen as a growth hormone almost. It really prompts the growth of the endometrium, and also prompts the growth of mammary glands to prepare for lactation. So estrogen levels increase, and that's going to prompt the proliferative phase of the uterus. So after this proliferative phase, you can see that it's not only the endometrium, but it's also the smooth muscle, that myometrium, that also gets thicker. And then the glands within the cervix are going to create more mucus to prepare, right, for in case we have implantation. So the mucus is sticky, and it allows it's actually the blastocyst that comes into the uterus. And then the last phase is going to be the last about 2 weeks of the menstrual cycle. and that's called the secretory phase. And that's where we really see a huge upswing in progesterone. So it's progesterone that really affects the secretory phase, because progestation, so now the uterus is preparing for implantation, and we are increasing the blood supply, the blood vessels are branching, we're getting engorged glands, we are starting to secrete glycogen-rich fluids, right? Because glycogen is glucose, it's going to help feed the embryo in those initial first stages of pregnancy. Cervical secretions get more sticky, forming a plug, and that's to isolate the uterus from microbes. So that's what's happening in the uterus. Now let's just take a look at the whole picture, looking at the gonadotropic hormones as well. So looking at number 3, the hormonal changes during the menstrual cycle. Overall, estrogen is first secreted from the granulosa cells of the follicles and then from the corpus luteum, right? That should make sense. So the first, we get estrogen from granulosa cells and also the corpus luteum. Progesterone is secreted from the corpus luteum. And then estrogens and progesterone feed back and inhibit our gonadotropins. There's one exception, and we'll talk about that in a second. So in the early to mid follicular phase, what we see here is a decline in plasma estrogen and progesterone, right? So I'm going to do a quick eraser right here. There we go. So what we see here is a decline in estrogen and progesterone, right? Because at the very beginning, that's what triggers menses. And because we have no more estrogen and progesterone, those two hormones actually negatively feed back, right, to the anterior pituitary. And so if we lose that negative feedback, if there's no more negative feedback, then we can actually go ahead and make more FSH and LH. So there's a decline in plasma estrogen and progesterone. LH and FSH consequently rise because there is no negative feedback. You can say it's not much of a rise, but it's a little bit of a rise. And they're going to then bind to receptors of the follicle. So FSH binds to receptors in the granulosa cells, causing the follicles to grow and then also differentiate into thecal cells. So follicle stimulating hormones, okay, FSH is going to stimulate the granulosa cells. And then the granulosa cells, some of them will mature into thecal cells. And the thecal cells are kind of like the Leydig cells of the male. So the thecal cells are going to respond to LH, luteinizing hormone. And then they're going to secrete your androgen. They're going to secrete testosterone. And the testosterone is going to diffuse into the granulosa cells, and the granulosa cells will then change testosterone into estrogen and secrete estrogen. Okay, so we've covered that first initial part of the early to mid follicular phase. As estrogen levels increase, they promote the proliferative phase of your uterus. Now, in the late follicular phase, the second half of the follicular phase, LH levels are going to rise, and they're going to rise dramatically, right? So we see this huge surge in LH. So at this point, it's interesting because normally your estrogen is going to negatively feed back to the LH and FSH levels. However, in this case, it doesn't. There's actually a positive feedback loop here. And the more estrogen that's being created will cause more LH to be made. And so more LH means more estrogen means more LH, and this is what creates that surge. So this is the exception where you don't have negative feedback, you actually have a positive feedback loop in this late follicular phase. Now, when we have this LH surge, we've already talked about, we have ovulation occur. So the enzymes are secreted, they're breaking down the follicular wall, and they're going to rupture about 18 hours after this surge. Then after the surge, estrogen's going to drop and progesterone levels are going to rise. So we can see a drop, right, of estrogen. Estrogen's basically done its job. It's going to produce this proliferative phase of the uterus, and it also produced that surge for ovulation. So we're pretty much kind of done with estrogen for the moment, and then progesterone's taking over. So the reason why progesterone is secreted is with the corpus luteum. The corpus luteum secretes this progesterone. And again, this progesterone is going to really influence the uterus to have the secretory phase, to prepare, to enrich for implantation. Because now that we've ovulated, there is a chance the woman's going to be pregnant. So we have to have this lining of the endometrium being really thick and nutritive. All right, so now moving into the luteal phase, right, of the menstrual cycle. Estrogen levels continue to decline, progesterone levels continue to rise, and again, prepare for implantation. And here there's a very powerful negative feedback where progesterone is going to inhibit your gonadotropic hormones. It's a power inhibitor, so progesterone is going to negatively feedback. So you can see how low the levels are, because when progesterone's high, it's going to negatively feedback on LH and FSH. And so then that's basically it. After 10 days, in absence of implantation, the corpus luteum starts to degenerate, and that's going to be our corpus albicans, our scar tissue. And then because of this degeneration, because the source of estrogen and progesterone go away, that's going to trigger our menses. Okay, so that's the overview of the menstrual phase, and this is a really good picture. It has everything overlapping with each other. I had to take this from the anatomy textbook. Your physiology textbook has separate pictures, but I like to have them all together so I can see how they overlap in time. And so this is a really good one to study because it really has everything here. And I wanted to mention I added an arrow because it's the uterus responds to what's the hormones provided by the ovary, right? So estrogen and progesterone are what's influencing the uterus. Now, I haven't talked about inhibin. Inhibin is also produced by the granulosa cells, and that's going to inhibit follicle stimulating hormone. Okay, so in the later phases here, right, in this luteal phase, we have an increase in inhibin and we have an increase in progesterone. Both of those are negatively going to feed back to both of our gonadotropic hormones, the LH and the FSH. So they're going to keep those levels down so that we don't have a second ovulation spike, and we can maintain the action of progesterone to keep the uterine lining thick. All right, so I know it's very complicated looking. I'm going to just clear that again. But it's something that you need to take time and look at and study. And one thing here, let's see. So I'm going to go back and just review a couple of things, but let's take a look at this picture. So this is just looking at that follicular phase. So remember that we talked about how the baby girls, the oogenesis is taking place in utero. So the primary oocytes are being created while the baby is still in the mother's womb. And so at birth, right, so this line is at birth. At birth, she already has all the primary all the primary oocytes that she's going to need for her entire life. And then this kind of sits there through adolescence. And then at sexual maturity, right, at puberty, that's when you get this follicle stimulating hormone, luteinizing hormone, and that's going to induce the maturation process of the follicle, right? So we're going to have a primary follicle, secondary follicle, our Graafian follicle, and then we have ovulation, right? So that after ovulation, the follicle becomes the corpus luteum, and if there's no fertilization, then it's going to degenerate into the corpus albicans. If there is fertilization, remember, it's the beginning placenta that secretes human chorionic gonadotropin hormone that's going to act on the corpus luteum so it does not degenerate. So it stays there and secretes estrogen and progesterone. All right, so taking a look at our histology slide, this is just a nice picture of our tertiary follicle, our antrum, the fluid in there, how large it is, and then our two coverings on top of the oocyte, the zona pellucida and the corona radiata. So quick thing about the birth control pills right here. So birth control pills are a mix of estrogen and progesterone, synthetic estrogen and progesterone, and they basically act on this hypothalamus pituitary axis. They're going to reduce the amount of LH and FSH release, and they're going to prevent ovulation. So birth control pills prevent ovulation. If a woman doesn't ovulate, there's no egg to fertilize, and there's no pregnancy. This picture is weird because I noticed that there is a pill missing right in the middle of the week. So I don't know what's going on there. So I grabbed a weird picture, but all right. So I think that is about it. Let me see if let's just take a look at the two phases again. So the follicular phase is going to promote endometrial growth. The follicular phase is going to promote oogenesis and follicle development, right? That's why it's called the follicular phase, because the follicle is developing. And inside those follicles, the eggs are developing. And we're going to have an increase to luteinizing receptors, an increase to progesterone receptors in the uterus, and an increase in luteinizing hormone secretion. In the pregnancy and late luteal phase, if there is pregnancy, we're going to have the estrogen here is going to promote duct growth. It's going to suppress lactation. It's going to increase prolactin, increase growth and contractile activity of the smooth muscle of the uterus, increase oxytocin receptors in the uterus, and promote fluid retention. That's why women who get bloated around the times of her period, this is due to estrogen. All right, so we're going to just look at this next couple of diagrams, and we're going to compare the commonality between the female and male hormone regulation of reproduction. So this is the female first. So this is a simpler look at what's going on. So the hypothalamus, right, that's going to secrete gonadotropin releasing hormone. That's going to act on the pituitary gland to secrete its hormones, FSH and LH. FSH and LH have two different targets. So follicle stimulating hormone is going to target the granulosa cells. The granulosa cells are going to help the oocyte develop. And they're also going to secrete inhibin. So inhibin is going to inhibit follicle stimulating hormone. Then LH, luteinizing hormone, is going to stimulate the thecal cells. Right, the thecal cells are going to secrete testosterone. So the thecal cells are really kind of like the Leydig cells of the testes. The thecal cells secrete testosterone, right? So the hormone, but testosterone diffuses out of the thecal cells, goes to the granulosa cells, where the granulosa cells convert that testosterone to estrogen and progesterone. Estrogen and progesterone inhibit both the hypothalamus and the anterior pituitary. So you can see the red line. So we have the negative feedback to the pituitary gland and negative feedback to the hypothalamus. There is an exception, right? The exception is that once each cycle, estrogen actually does not negatively inhibit the gonadotropin releasing hormone. There actually increases the luteinizing hormone being produced. So it's going to create that LH spike, right? So we're going to actually have positive feedback here around ovulation and get that LH surge. That's the exception. For males, coming back to males, the hypothalamus, same thing. It's going to secrete gonadotropin releasing hormone. We have two hormones so gonadotropin releasing hormone is going to cause the pituitary gland to make FSH and LH. FSH is going to stimulate the Sertoli cells, and the Sertoli cells are going to help with spermatogenesis, right? So really similar, because the Sertoli cells are kind of like the granulosa cells, right? The granulosa cells respond to FSH by maturing the oocyte. The Sertoli cells respond to FSH by maturing the sperms, the spermatogenesis. They both, Sertoli cells and granulosa cells, will secrete inhibin, and that's going to feed back on its own loop. So it's going to feed back to inhibit FSH. Looking at luteinizing hormone, LH is going to induce the Leydig cells to secrete testosterone. And the Leydig cells are kind of like the thecal cells, right? So they're both responding to LH. They're going to both secrete testosterone. But in the case of females, the testosterone gets converted to estrogen and progesterone. In the male, it stays testosterone, and it negatively feeds back to both the pituitary gland and the hypothalamus. Just like the female, estrogen and progesterone negatively feeds back to the pituitary gland and the hypothalamus, with the exception of ovulation. All right, so that is the female reproductive menstrual cycle. Some wrap-up questions to think about. So which of the following structures is described as a canal measuring 8 to 10 centimeters in length that connects the cervix with the external environment? The answer is vagina. Which of the following is not a function of the granulosa cells? So you can pause it to think about it. Okay, D is your answer. It's not a function. During which phase of the uterine cycle does uterine tissue renew itself in preparation for possible pregnancy? The answer is proliferative phase. All right, and that's the end of this part.